Competent cells are bacterial cells that are specially prepared to take up foreign DNA from their environment, like plasmids. This ability to take in external DNA is crucial for genetic transformation, a process where new genetic material is introduced into an organism. Whether you're working in molecular biology or genetic engineering, competent cells are fundamental tools. Let's explore what makes them so essential and how they work. There are two main types of competence in bacteria: natural competence and artificial competence. Natural competence. Some bacteria, like Streptococcus pneumoniae and Neisseria gonorrhoeae, have the natural ability to take up DNA from their surroundings. This process is built into their life cycle, and it allows them to incorporate new genes into their genome. Natural competence contributes to genetic diversity and helps bacteria adapt to changing environments. For example, when bacteria take up genes that provide antibiotic resistance, it increases their chances of survival in a harsh environment. In these naturally competent bacteria, specialized proteins bind to the external DNA and help it pass through the cell membrane. This natural process happens without any external intervention, but it's limited to certain species of bacteria. Artificial competence. However, most bacteria, like Escherichia coli, E. coli, are not naturally competent. However, scientists have developed artificial methods to make these cells take up foreign DNA. Artificial competence is critical because it expands the range of bacteria that can be genetically transformed. There are two common methods used to create artificially competent cells. 1. Chemical competence. In this method, the bacterial cells are treated with a solution, like calcium chloride, which temporarily makes the cell membrane more permeable to DNA. This is often followed by a heat shock, a brief exposure to high temperature, which helps the DNA move through the membrane into the bacterial cell. It's a relatively simple and widely used technique in molecular biology labs around the world. 2. Electroporation. This technique involves applying a brief electrical pulse to the cells. The pulse creates tiny pores in the cell membrane, allowing the DNA to pass through. Electroporation can be more efficient for certain types of cells and DNA, but requires specialized equipment. It's used in situations where high transformation efficiency is needed. Why competent cells are crucial for genetic transformation. 1. DNA uptake efficiency. Normally, bacterial cell membranes act as a strong barrier to foreign DNA. Competent cells, however, increase the likelihood that the bacteria will take in external DNA, such as plasmids, circular DNA molecules that can carry specific genes, like antibiotic resistance or genes of interest. 2. Genetic manipulation. Competent cells are essential tools for genetic engineering. By introducing plasmids into bacterial cells, scientists can clone genes, produce proteins like insulin, or create genetically modified organisms, GMOs. These transformations allow bacteria to express new traits, which can be critical for research and commercial applications. 3. Antibiotic selection. After transformation, scientists often use antibiotic resistance genes to identify the cells that successfully took up the new DNA. Only the bacteria that have incorporated the plasmid will survive on plates containing antibiotics, making competent cells vital for this selection process. In summary, competent cells, whether naturally occurring or artificially prepared, are key to advancing genetic research and biotechnology. They enable scientists to manipulate DNA, unlock new capabilities in bacteria, and drive innovations in medicine, agriculture, and industrial biology.